Hi, everyone. Welcome to your Chapter 4 lecture. And if you're viewing this lecture during the fall of 2020, then in order for you to be on pace with your course lectures, you should be viewing this on Monday, August 24th. If you're viewing this later, then please know you're a little bit behind where you need to be in terms of watching video lectures, and you're gonna need to speed up the pace, okay? Now, before we jump into the meat and potatoes of today's lecture, I wanted to show you some additions that I've made to Blackboard to make your life and studying and navigating your e-textbook a little bit easier. So I want you to notice this is your home page, and I want you to go to the content page at some point in the next week and notice that I've made some changes to the folders that are available to you. Before, we had a syllabus and general information folder, a week 1 folder with all of the content for week 1. And by now there's a week 2 folder that's posted that contains this lecture as well as other associated material. But you'll also notice that I've added a folder called textbook stuff. And I added this after a class meeting on Friday when a student pointed out that there was no way for them to get to the e-text only. They could get to my assignments, but they couldn't get to the e-text from Blackboard. And so to make that easier for you, I didn't realize that. I'm still learning this platform. So what I've done is I've added some links here, okay? Now this first link, I'm not going to click on it. This Welcome to Mastering Biology link will take you to like a master overview page for all of the resources and assignments associated with the Mastering Biology that you purchased. So you can get to anything that is associated with your purchase by clicking here. I'm not clicking there because I think that my instructor homepage looks quite different than the student homepage. But I will tell you that if the student homepage looks anything like my instructor page looks, then it's really busy and a little difficult to navigate. So I went ahead and put these 3 links on here thinking these might be a little easier for you, okay? This first link is going to take you directly to the Pearson e-text. So let me show you. If you were to click on that, I've already pulled it up for me, but if you click on it, what you would find, let me go back here. Hold on. You would get a picture of the cover of the textbook, I hope. Let's see. Come on, slow internet. Yes. So what we should get here in a second is a picture. Okay, this is one of the pages. You can navigate through the pages, like flipping a page by going back and forth using these buttons. So if you wanted to just flip pages, that's what you do. But an easier way to navigate things is to go to this hamburger menu in the upper left-hand corner, click on that, and what you'll get is an outline of the textbook. It's basically a table of contents, so you can jump to wherever it is you would like to be. So let's say that you wanted to study chapter 1 because you've watched that video and you wanted to look at the text, you could click here. If you wanted to go to a particular section of that chapter, you would just click this button here. And you could go to various sections, okay? So if we click on this, go to chapter 1, one of the options that we have is to go here and use some of these different apps, or whatever you want to call them, different add-ons. You can use a notebook here to take notes if you want to do that. One of the apps that I like is their study app. So for each chapter, if you click on study, it goes through and it defines all the vocabulary for that chapter for you. And at the very top, you have some different ways to use that vocabulary. You can do flashcards, you can play a matching game, or it'll ask you questions and you can fill in the blank. So that's one way you can navigate your e-text. All right, let's go back to Blackboard. So that's what that link will do for you. I'm going to skip this link for just a second, and I want to draw your attention to this last link here where it says study area. If you click that, then what you should get is this here. I'm going to go to the home right quick so you can see what it looks like when you first get to this page. There we go. When you first get to this page, when you first click on the link, this is what you'll see. You have all these options. You can go directly to the e-text from here. You can also look at all the animations and what have you. I think it's quite useful to look at things by chapter. So if you click on that, You can see all the chapters that we'll go over. And let's go and look at chapter 1 again. Again, you can go straight to the e-text from here if you like. You can also look at any videos or interactive figures that they have. And here you can even take a practice test in order to figure out how much you remember about the chapter. All right, now let's back up and look at this one. This Mastering Assignments link is the same one that appears also here, okay? Same link. And if you click that, what it does is it will take you to a master page that shows you a list of every single thing that I've made available to you as an assignment in this course. Let me show you what I mean. So let's pretend we've clicked that. I've already brought it up. This is what you would get. Don't panic, okay? So this is the homework I've already assigned and you're familiar with, okay? You can see that you've got a due date here of August 28th, which is this coming Friday, right? We're all familiar with what this homework is. It's a very easy, very simple assignment to get you used to using this platform. It is the easiest grade you will make in this course, so you better take advantage of it. Everything from here on out will be a much harder grade, okay? But then you also see all of these crazy dynamic study modules that are indicated with this little flame icon here. And you can see that I've got a due date here. Now, these are not graded at all. They are associated with 0 credit. And the only reason that I gave them a due date of September the 2nd is because that is the Wednesday before your first exam on September the 4th, okay? So ideally, you would have completed these before then. However, these will be available throughout the duration of the course. Now, this page to me is very busy. So let me show you another easier way to get to this information, okay? If you go to the week 1 folder here, so this should look familiar. This was already here. The week 1 PowerPoints were here, the video lectures, and the videos of the class meetings. Incidentally, the PowerPoints are just the PowerPoints that I use when I do my video lectures. They are only there if you choose to download them in order to take notes, okay? There's nothing else you need to do with those. All right, so all of these things were already there. But what should be new to you is this homework tab and this study aids tab. And if you click on the homework tab, what it does is it's a link to just the homework assignment, okay? If you click on this tab, you get this, okay? And it basically takes you right to the homework and you can launch it and you can, if you were to click here, you would start with this first question set, et cetera, et cetera, et cetera, okay? And you can see the due date, you can see how it's being graded. Lovely, okay? Let's go back. If you click on study aids, that's where those dynamic study modules are listed. And what I've done is I've posted all of the study modules that are associated with the lectures for this week. And I've given you 2 question sets per chapter, okay? These are here solely for your use. I will not grade them. I don't care if you do them or not. However, I will tell you, they're super useful, okay? If you go to this link and you take their little practice one, it shows you that what they do is they ask you a series of questions. And when you go to answer, you have the option of saying, you know, clicking an answer and saying, I'm confident about this answer, or you can click an answer and say, I'm not confident about this answer, or you can simply answer, I don't know. And if you don't know or you're not sure, what it will do is it will come up with information to help you answer that question, and then you move forward and answer some more questions, and it'll circle back around to the things that you were unsure about until you have a complete understanding of the chapter, okay? The only thing I will say that's a negative is you guys know that occasionally there are sections of chapters that I leave out, and I really couldn't edit these to leave out the stuff I'm not talking about. So just to reiterate, you are only being tested on the things that I cover in lecture. I still think that these are worth your time to look at, but just understand that there may be more information here than what I ordinarily talk about, all right? So hopefully all of those things are useful to you as you navigate through the course moving forward. Now, let's move on to talk about the meat and potatoes of, let's see, I want to use this one, the meat and potatoes of today's lecture, which is talking about chapter 4 and nucleic acids, okay? So you guys know that we are in a unit where we are discussing macromolecules. I'm just gonna put macros up here, right? And if you are here at lecture 4, then presumably you have already watched the first lecture video on the first macromolecule we'll discuss, which is proteins, right? So today we're going to move forward and talk about a second class of macromolecules called nucleic acids. Now, when we discussed proteins, you recall that there was just a huge variety. There was so many different types of proteins that you could have, right? Based on different combinations of amino acids. But when we talk about nucleic acids, they really only come in 2 different flavors. You can have DNA as a nucleic acid, or you can have RNA as a nucleic acid, okay? What's the difference? DNA contains a sugar called deoxyribose. I'm just going to put deoxy here, while RNA contains a sugar called ribose. And if that isn't meaningful to you, that's okay. It will be by the end of this lecture. So these are still macromolecules. These are still very, very large molecules, as you'll see, but there's just 2 different types, DNA and RNA, whereas we have a variety of proteins, okay? So key concepts that I want you to know as we move through this chapter. I want you to know what constitutes a nucleic acid. I want you to understand the structure and function of DNA, the structure and function of RNA. And then at the end, we're going to discuss a little bit about whether life might have evolved from RNA. And one thing I have for you down here in your notes is that by the end of this chapter, we will have discussed 2 theories for how life might have begun on this planet. And so you should be able to discuss those 2 theories at the end of this lecture, okay? All right. And when I say by the end of this chapter, 2 theories, one of those theories was in the previous chapter, just to clear that up. One will be in this chapter, one is from a previous chapter. Okay, nucleic acids. Let's talk about them. So, as I said earlier, we are talking about macromolecules, and you know that many of our macromolecules are polymers, right? They are many unit type molecules that are made up of individual units called monomers. Now, if we were talking about proteins, the polymer would be the protein, right? And the monomer would be amino acids. I'm just going to put AA. But today we are not talking about proteins. Today we're talking about nucleic acids. So the polymer is going to be either DNA or RNA. And the monomer that we're going to be talking about that makes up these polymers are called nucleotides. Okay, now a nucleotide, whether it's for DNA or RNA, is made up of 3 parts, okay? Part 1 is going to be a sugar molecule, okay? Attached to that sugar molecule is going to be part 2, a nitrogenous, meaning containing nitrogen, base. And part 3 is going to be a phosphate group. Now, I told you that DNA and RNA differ in the type of sugar that they contain. So they have different types of nucleotides that have different sugars, and then you'll also see that they can have different bases. But this is the fundamental structure of an individual nucleotide. Now let's get a little more detailed. Let's start out and draw a nucleotide that would be one that would go with RNA. Okay, so that means it would have a ribose sugar. The ribose sugar starts out with an oxygen, okay, attached to a carbon, another carbon, another carbon, carbon here, and then a 5th carbon here. So it is a 5-carbon sugar. And by convention, we number these sugars starting with the oxygen and going clockwise, and we say that this is the 1', 2', 3', 4', and the 5' carbon, okay? It is important that you know this numbering because that will be important when we learn about the structure of both DNA and RNA a little later. Now, I said that I was going to show you ribose, which is the central sugar in RNA. And so in order for me to show you that, let's first start out, and I'm going to put just a circle that says base here, okay? Because it turns out the base is always going to be connected to the 1' carbon, but if you are RNA, the bases can come in 4 flavors. It can be adenine, cytosine, guanine, or uracil, okay? Now that might seem a little weird to you because you might be familiar with DNA where you have adenine, cytosine, guanine, or thymine, right? You guys know that DNA has A's and C's and G's and T's. And yes, you need to know what the A, C, G, and T stand for. But one difference between DNA and RNA is this right here. Instead of having thymine, RNA has uracil, but everything else is the same, okay? So you would have one of these 4 bases in an RNA nucleotide, okay? And it's always going to be attached to the 1' carbon. Then when you get to this 2' carbon, if we're talking about RNA, you're going to find an OH group. Similarly, when we get to the 3' carbon, you would find an OH group, okay? 4' carbon doesn't have anything I want you to know about, but the 5' carbon is always going to be where the phosphate group is attached. Hang on, I'm gonna look something up really quick. Yes. Okay, so that's where your phosphate group is going to be attached. And the phosphate group would look something like this. It would be, let me do a little erasing here so that you guys can see my drawings a little bit better. All right, so it's going to have an oxygen, a phosphorus, an oxygen, and there's a double bond here, and then I think there's another oxygen there, okay? That constitutes a phosphate group. I rarely draw out the phosphate group. Oh, sorry. I rarely draw those out, but just so you know that that's what a phosphate group looks like. So this whole thing is actually called a ribonucleotide. Okay, now let's draw a deoxyribonucleotide. I'm going to abbreviate nuke. Okay, so I'm going to draw you a deoxyribonucleotide. It's going to have deoxyribose as its sugar as opposed to ribose. So let's start out again with the sugar molecule. Now this time I'm going to draw it a little bit differently. I'm not going to put the C's in. When you get to organic chemistry and you start drawing shapes like this, it's kind of, the carbons are understood to be there. So if you've got a little corner here, 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 it's a corner here and there, it's just understood that the carbons are there. So this is still the 1 carbon, 2, 3, 4, and 5. But to make this a cleaner drawing, we're not going to actually write out the letter C. So just like with a ribonucleotide, your bases are always attached to the 1' carbon, okay? And your base can be either an A, a C, a G, or a T for adenine, cytosine, guanine, or thymine, okay? Now, here's where we get the difference. Recall that if we are ribonucleotides, if we are going to be part of RNA and the sugar is ribose, there's an OH group on the 2nd carbon. In deoxyribose, it doesn't exist. Deoxy, without oxygen, we've lost an oxygen. Now, what does exist is the OH group here, and that's going to be really important. This OH group on this 3' carbon is actually going to be involved in linking up the various nucleotides. That's true for ribonucleotides and deoxyribonucleotides. So it's important that you understand that. And then the phosphate group, which I'm just going to draw as a P and a circle this time, is still going to be attached to the 5' carbon, okay? So the base is always attached to the 1 carbon. You may or may not have an OH group here, depending upon whether you are a ribonucleotide or deoxyribonucleotide. You have an OH group at the 3' end and a phosphate at the 5' end. If you aren't drawing this out for yourself for both versions, for both RNA and DNA, then you are not studying effectively, okay? Because I will ask you about it. All right, now let's move forward. So here's a little bit cleaner version of what I just showed you. You've got a 5 carbon sugar. It's got the numbering system here for you, okay? You can see that the nitrogenous bases are always attached to the 1st carbon. The phosphate's always attached to carbon 5. If you are RNA, you have a ribose with an oxygen there. If you are talking about DNA, that oxygen is absent because now we have deoxyribose. Now let's focus in a little bit on the bases, okay? Remember I told you that you could have, if you were RNA, you could have A, C, G, or U. If you're DNA, it can be A, C, G, or T, okay? This diagram has them divided up for you, okay? And it has them divided into 2 categories. The pyrimidines, which have 1 ring on their structure, and the purines, which have 2 rings on their structure. Now, you probably remember from high school biology when you were learning about DNA that A always pairs with T and C always pairs with G, right? Okay, well, what we really want you to understand at the college level is that a pyrimidine always pairs with a purine. Okay, and you'll see why in just a second. Now, how are you going to remember which bases are pyrimidines and which bases are purines? Well, I have a couple of mnemonic devices that will help you remember which one is which, okay? I remember it using this one down here in your notes. I say pure silver, okay? Pure for purine, silver, well, I know that the chemical symbol for silver is Ag, and Ag are the initials for adenine and guanine. Now that might not be helpful for you if you don't know chemistry, so here's another way of doing it. Here's a sentence, climbing the pyramid, George avoided 2 pursuers. Okay, climbing the, that's a C and a T, right? And then pyramid for pyrimidine. C, T, pyramid, pyrimidine, okay? George avoided 2 pursuers. George, G, avoided 2 pursuers for purines, okay? So if that is helpful for you, then great. So I may ask you about whether or not, maybe I'll give you a diagram like this that maybe has 1 ring or 2 rings, and I might ask you things like, is a purine or pyrimidine attached to this sugar, okay? Now what I don't want you to have to do, you don't have to be able to draw these out. I just want you to know that cytosine has 1 ring and guanine has 2. You don't have to actually draw the chemical structure, okay? All right, moving forward. Let's talk a little bit about how these individual nucleotides polymerize to become a polymer, okay? So remember, polymers are made of monomers. So far, what I've told you about is the monomer, okay? We haven't even gotten to the polymer yet. So let's get there. All right, here we have 1 nucleotide, and here we have another nucleotide. And I want you to ask yourself whether what I am drawing or what I am going over is going to form a DNA or an RNA molecule, okay? I'm not going to tell you, but I want you to figure out whether or not that is true. Also, ask yourself, is this a purine or a pyrimidine? Is this a purine or a pyrimidine? Make sure you can answer those questions. But in the meantime, what I want to draw your attention to is this 3' OH group that I told you about and this 5' phosphate group, okay? So what's going to happen is, you see we have a hydrogen on this 3' OH group, and it's going to bond with an OH here. See how this is negative? So it's going to draw a hydrogen from the solution, the water solution around it. Okay, so you're going to take a hydrogen from here and an OH from here, and that's going to end up being H2O. So these will then merge together, right, like thus, okay? So here we have a bond that is formed and water has been lost. This is your condensation reaction, okay? And of course, they've written it for you right there. Now, this type of bond has a particular name. It is a phosphodiester. I've written it for you here. Phosphodiester bond. That is the name of the bond that links two nucleotides together. Doesn't matter if you're talking about RNA or DNA, okay? And let's contrast that with proteins. What was the name of the bond that formed proteins? See if you can answer that for yourself. Okay, we'll talk about that in class. So once we have this beginning of a polymer, you can see that this polymer has an orientation. It's got a 5' end that has a phosphate on it, and it has a 3' end with an open OH group where you could bring another nucleotide in and add onto this chain, right? So I always like to think of this 3' OH group as sort of the parking spot for the very next nucleotide to come in and add to this chain. And what you would end up with over time is what we call a sugar phosphate backbone on one side. And hopefully you could see that phosphate, sugar, oops, here, phosphate, sugar, right? So we've got a sugar phosphate backbone or phosphate sugar backbone on one end and the bases on the other, right? So you've got one side with the backbone and one side with the bases facing inward, okay? Again, check yourself. Is this RNA or DNA? Okay, moving on. I want to switch and talk specifically about DNA now. So I told you that we had two different flavors of bases. You could be a pyrimidine or you could be a purine, and that purines always pair with pyrimidines. I also told you that DNA, if you have bases in DNA, you can have A, C, T, or G, right? Okay, so let's take a look at, let's start here at this C diagram here. I want you guys just to look at this half first. So this is kind of what I've already shown you. What you have is a sugar phosphate backbone, and we have bases facing the inside. And one of the reasons it's important for the bases to face inside is because they're going to interact with complementary bases, following what we call complementary base pair rules. And you guys know those. I mentioned them on the last slide. You know that A pairs with T and C pairs with G, right? Okay. So let's look. Here we have a G and a C. That seems right. GC, AT, AT. Now I want you guys to look really closely at this diagram. A couple things I want to point out. For DNA, you always have two strands, okay? That is not going to be true in RNA. So we've got one sugar phosphate backbone here and another sugar phosphate backbone here. And then we have the bases lined up with a purine and a pyrimidine each, and they have associated using base pair rules. Another thing that I want you to notice is that on this particular side, on this strand of DNA, we have one end where the phosphate's on the top and the OH is on the bottom. So this strand is oriented from the 5' carbon to the 3' carbon, okay? Now let's look at the second strand of DNA. It's oriented upside down, isn't it? We see the 3' end on top and the 5' end on the bottom. And the reason that it's flipped upside down is because that's how you can make these things, the bases, line up properly so that they are associating properly. So we call this configuration anti-parallel. DNA consists of 2 strands, and those 2 strands are anti-parallel. And again, the reason that one is upside down relative to the other is so that these bases link up properly. Now let's talk about how these bases link up. You can see that there's some dotted lines that are drawn here, dotted and highlighted. That's actually indicating for you that these are not covalent bonds. These are in fact hydrogen bonds, okay? Meaning that they are not permanent, okay? So hydrogen bond here, hydrogen bond here, here, here. Hydrogen bonds, hydrogen bonds. Notice that if you are talking about the guanine-cytosine association, you can make 3 hydrogen bonds. If you're talking about the adenine-thymine association, you can make, you can make 2 hydrogen bonds. But what I want you to understand is these 2 strands of DNA, this strand here and this strand here, they're stuck together via hydrogen bonds, but they're stuck together like magnets. And magnets can be pulled apart. So we're going to be able to pull this strand apart from this strand, all right? So that's one feature that we're going to talk about because we're going to talk about replication in just a second, all right? So I wanted to move to this one last. Remember how I told you that purines and pyrimidines always associate with each other? Notice that in this space here, we've got 1, 2, 3 rings, 1, 2, 3 rings, etc., etc. And it's the 3 rings together that create the perfect amount of space between these 2 strands of the DNA molecule. If you had an accidental pairing of, say, 2 purines together, okay, then you would end up with not enough space in your DNA molecules. As a matter of fact, what you end up with is a backbone that has a bump on it, like this. And that's one way your cell knows if you've messed up your DNA. It can come along and feel this bump and cut out this faulty bit, okay? What if we had 2 purines in this space? Well, that means we leave a gap, and what we'd actually find is a dip in the DNA backbones like this. And again, there's enzymes that would come along and actually feel that there was a dip, and they would cut this out because that would be an obvious mistake. So in order for the backbone to line up properly, you've got to have 1, 2, 3 total rings, okay? 1 from a pyrimidine and 1 from a purine, all right? Now, a couple of things I want you to think about for when we get together and chat during our live class session. So I told you that magnets can be pulled apart. So what are some other ways, since this we know is not a permanent bond, how do you think we might be able to separate one strand of DNA from another strand? What are some ways that we might be able to pull them apart? And are there any things that we can do to make this molecule more stable? Okay, those are questions that I will be discussing as we go through our class discussion. All right, moving forward. So just like proteins, DNA has a primary structure, a secondary structure, and in another slide we'll talk about a tertiary structure, okay? So remember that with proteins, the primary structure was just the sequence of amino acids, right? That was for proteins. Similarly, the primary structure of DNA is just the sequence of bases, right? So this would kind of be like the primary structure, just a flat TGCTT, okay? But what happens once you get these guys together, because of the way the molecules are configured, it starts to twist on itself to make the secondary structure of DNA, which is called a double helix, okay? It looks like a spiral staircase. This configuration was actually discovered in 1953 by two folks named John Watson and Francis Crick, and we're going to talk a little bit more about the discovery of DNA when we get to a future chapter on DNA synthesis. Actually, I should say on the discovery of the structure of DNA. Okay, just like I told you earlier, we still have this anti-parallel configuration. See, 5' here, 3' here, and then the other one is flipped upside down, right? So we've got 5' here, 3' here. So even once we start doing our spiral staircase configuration, you've got 5 and 3 and 3 and 5, okay? If we were to look at a more accurate diagram, I guess you could say, that was just a little bit more detailed, you would see some other features. For example, DNA has a major groove and a minor groove, and this tells you the distance between bases. I'm not so interested in you knowing that. We'll discuss that in genetics if you ever get to my genetics course. But for now, just understand that the primary structure of DNA is the sequence of bases, like what you see here. So this is the primary structure. And the secondary structure is what you see here with the double helix. Now, does DNA form a tertiary structure? Now remember that in proteins, you had a primary structure that was like beads on a string, right? And then in your secondary structure, you could have it pleat up like a sheet, or you could have it roll up like a telephone cord. That was in proteins. And then proteins, the telephone cord or the sheet could knot up into a three-dimensional shape to form your tertiary structure. Well, we got kind of a similar thing going on with DNA, don't we? So you have your sugar phosphate backbone and your bases, right? And that represents your primary structure. Oh, golly, yeah. I'm just drawing stuff. And then it'll twist in on itself like a double helix to make the secondary structure. And it will sometimes knot in on itself to make a tertiary structure. So here's a couple of examples of that. In bacteria, you only find one circular chromosome. We're going to talk about that a little bit more later. You know how you have multiple linear chromosomes. Bacteria have one, and it's a circle. And so in order to kind of make sure that it's out of the way in the cell and it's not taking up too much space, it undergoes something called supercoiling, where it twists in on itself and it forms this supercoiled knot. All right, so that's the tertiary structure of DNA in bacteria. So I'll put that here, tertiary in bacteria. Okay, let's talk about things that are what we call eukaryotes. And if you don't understand that term, you will soon, but I will just tell you that you are a eukaryote because you have a nucleus and you have multiple linear chromosomes. And you actually have a bunch of DNA in your cells. If you could actually take your DNA and pull it apart into a tiny, very, very thin molecular thread, it would be something like, I don't know, 6 feet long. It's a crazy, crazy amount. In each nucleus, it would be that much, okay? So in order to organize it and in order to wrap it all up so that it fits inside your nucleus, the tertiary structure of DNA in eukaryotes is that it is wound around other proteins called histones, the same way that you would use a spool to wind thread around, okay? So if you were winding thread around a spool, that's exactly how these histone proteins organize DNA, okay? So the DNA is wound around histones so that it can be condensed and fit inside of your nucleus, all right? So that's the tertiary structure in eukaryotes. Okay, it's wound around histones. All right, let's move forward here. Let's see here, what do we have? Okay, now, I told you, come on, there we go. I told you guys during our chapter 1 lecture that one of the features of life is that life has to be able to store and use information. And we all agreed that where that information is stored is in your DNA. And if you are a multicellular organism, you have to be able to take that information, copy it, and then give it to the daughter cells as you make more and more cells to become a multicellular organism. All right, so how does that work? Well, the neat thing about our DNA is that it's a mirror image, right? You've got one strand that has the complementary base pair from the other strand. So if you separate these strands, you can end up with a template such that you have an old strand and a new strand and an old strand and a new strand. Let me see if I have a better image of that. No, I don't. Okay. So essentially, you have, I'm going to draw it linearly. Let's say that you have an A, C, T, G, boop, boop, boop. And then you would have T, G, A, C. And if we wanted to copy this information, what we would do is we would disrupt the hydrogen bonds that are holding this together so that we had a template strand here, A, C, T, G, and a template strand here, T, come on, T, G, A, C. And then we would just use these to make a new copy, boop, 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 and we would end up with two identical strands. One strand would have one old and one new, and the other strand would also have one old and one new. So DNA forms a template for its own synthesis, which is really nice. And we're going to talk in detail about how this process happens. I have some questions down here. Which carbon is the phosphate group attached to? Okay. Yeah, answer these questions for yourself. In this image, which carbon would the phosphate groups be attached to, and which carbon is open for additional bond formation? And you would know that by looking at whether you had a 5' or a 3' end. Okay, let's switch gears and talk for a second about RNA. Okay, so RNA is very similar to DNA in that it has a primary, it actually has a secondary, and it can have a tertiary structure. Now, RNA is different from DNA in that it is single-stranded, which is often abbreviated SS, okay? So it doesn't have two backbones with complementary base pairs in between. It just has this single sugar phosphate backbone with individual bases attached to it, okay? So this sequence right here, without a second side, represents the primary sequence for this RNA. It's just the sequence of bases. And unlike DNA, the primary structure of RNA is single-stranded. All right, so, but a couple things to notice. Just like with DNA, we have a 5' end and we have a 3' end. Down here is where you could add nucleotides to create a longer chain, all right? So let's look at the secondary structure of RNA. Now, because RNA is single-stranded and it's got these base pairs sticking off, the bases, the RNA and DNA bases, with the exception of uracil and thymine, are the same, and they follow base pairing rules. They are attracted to one another. So even though RNA is single-stranded, it's still, A is still attracted to C electromagnetically, and instead of T, U is still attracted, no, sorry, I said that wrong. A is still, let me go. C is still attracted to G, and U is attracted to A, okay? So it's single-stranded, but what happens is sometimes that single strand will collapse in on itself because you can see G wants to hang out with C and A wants to hang out with U. So this is a single strand, but it's kind of formed some self-associations here. And one common shape that results from these self-associations is called a stem and loop shape. So can you see you've got a stem here where you're starting to get a little bit of a double helix and a loop here. But notice this is still a single strand of RNA. We can follow that up, boop, 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 and follow it back down, okay? So this stem and loop is an example of a secondary structure, but we are still single-stranded, okay? Now let's look at what tertiary structure might look like in RNA. Sometimes what happens, so here's a flattened out version of our stem and loop. Remember, we're still talking about single-stranded RNA, but sometimes this stem and loop will start to fold over onto itself until it forms a three-dimensional shape. And that three-dimensional shape is called a pseudoknot, okay? So this is the tertiary structure of RNA. This isn't always how you find RNA, but you can definitely find it in this shape. Now let's kind of summarize some of the things that I've talked about. I really like this table, this compare and contrast table. And when I really like something, it usually means that you probably should pay attention to it, okay? So let's summarize some of the things that I told you. The primary structure of DNA is the sequence of deoxyribonucleotides, okay? But I always think of it as being the two-stranded version versus the RNA's primary structure is the sequence of ribonucleotides, but it's the single-stranded version, all right? Secondary structure is our two anti-parallel strands that twist into our helix, our double helix, okay? The most common secondary structure that you would find in RNA is called a stem and loop, okay? Tertiary structure, you can either have in DNA supercoiling, sounds good, supercoils, or compact structures that are wrapped around histones, okay? In RNA, you can have a variety of knotted structures, but one that I told you about was called a pseudoknot. That's your tertiary structure. So I really like this one. I would very much anticipate a question from me that says, list the differences between RNA and DNA. This is the difference in their primary, secondary, and tertiary structures, right? But don't forget that some of their other differences include the double-stranded versus single-stranded, the use of thymine versus the use of uracil, and the use of deoxyribose as your central sugar versus the use of ribose as your central sugar. All right. Now, I told you that the function of DNA was information storage, right? So let's go back here. The function of DNA is information storage. And the nice thing about it is we can store and copy that information because we have a template. We can just pull this apart and make more and more Xerox copies. But RNA has a little bit different function from DNA, all right? And I want to tell you about 3 of those functions. Okay, the first function is that RNA acts as an intermediate between DNA and proteins. Intermediate, all right. So this diagram here to the left should look quite familiar to you. This, if you've watched chapter 1, is the picture that I use to describe something called the central dogma. Okay. And in the central dogma, you know that DNA gets transcribed into RNA. Now, why is that? Notice that the DNA in a eukaryote is living inside of the nucleus, and it doesn't leave the nucleus. It stays there all the time. And that kind of makes sense if you think about it. If this is the instruction book for how to run the cell, you want to protect that instruction book, because if you damage that instruction book, then you're in trouble, aren't you? So DNA doesn't ever leave the nucleus. Instead, it gets transcribed into messenger RNA, which can leave the nucleus. So here it goes, out of the nucleus, boom. RNA has now acted as an intermediate between DNA and then the proteins that it will later be translated into. So that's the first function of RNA. The second function of RNA is that it acts as a template for protein synthesis. So let's get back here. This messenger RNA is going to serve as a template to make proteins. And here you have your messenger RNA. And what you can see is that sections of this messenger RNA pair up with another type of RNA that is attached to an amino acid. And so we can use this feature, this section matching this section, matching this amino acid, to link amino acids together to make a protein. So can you see that this is functioning as a template for this, okay? And so this is a process called translation. We're gonna get into great detail about that later in the semester. So function one is that RNA can be an intermediate between DNA and the protein. It acts as a messenger. Two, it's a template for protein synthesis. And the third thing that RNA can do is it can catalyze chemical reactions. Now, you may be thinking to yourself, what? This is function three. In the protein lecture, I told you that enzymes, which are a type of protein, enzymes, they function as catalysts for chemical reactions. Now, what does a catalyst do? What a catalyst does is it speeds up a chemical reaction without itself being consumed in the reaction. So it's not actually part of the reaction. It just acts as like a little bowl where the reactants can sit and interact with one another, and then out pops a product, okay? And because these catalysts are shaped just right for the reactants, it helps bring them together and speed up the rate of the chemical reaction. Typically, catalysis of chemical reactions is mediated by enzymes, but it turns out that RNA can also fold up on itself to form a knot-like structure, as I just told you. So this is a tertiary RNA structure. And when that happens, it looks a little bit like a protein. And so it starts functioning a little bit like an enzyme, but it's not a protein, so we're going to give it a different name. We're going to call it a ribozyme. So RNA can function like enzymes do, only we're going to call it a ribozyme instead of an enzyme because it's made of a nucleic acid, okay? So one of the questions that people have asked about is whether or not RNA could catalyze its own synthesis. And you'll see why in a second. Can it catalyze its own synthesis? Meaning, could you have a strand of RNA that folded up on itself and made more strands of RNA? So could it make itself? Well, it turns out the answer is yes. And this is one reason that we think that life may have begun on this planet with RNA self-replicating. And here's how that might have worked. So let's say you have a single strand of RNA and it's floating in a primordial pool where there's a bunch of just free bases floating around, okay? So these free bases float around and they line up according to base pair rules, okay? So now you have an original RNA strand and a copied RNA strand, and then they split apart and you have two copies, okay? So this wouldn't happen with DNA so much because DNA is quite stable, but RNA, we do think through the use of ribozymes as the enzymes that cause this to occur, that ribozymes could have made a bunch of RNA, okay? Now let's think about this. If you have ribozymes making a bunch of RNA, then what you've done is you've satisfied 3 of the 5 characteristics that we laid out for life, okay? So if that's true, then your ribozymes, they contain information, because your ribozymes have a series of bases, right? So here you go right here, a series of bases that act as information. Okay, ribozymes can cause the RNA molecules to replicate. And if there were any mistakes in that replication, then you would end up with evolution. So the only two features of life that we're missing are cells and the energy that cells need to consume. So there's a lot of hypothesis that what might have happened is that life might have begun with a ball of RNA that started replicating itself, and that over time some lipids gobbled that RNA up to make the first cell. All right, so that ends our lecture for chapter 4. Hopefully you found all of the resources I posted for you helpful, and if you have any questions, we can discuss them during our class meeting. Talk to you soon. Bye-bye.